Well, thank you. Thank you everyone for coming and for having me. I'm super excited to be here. So my dog's named after Oliver Sacks, who is a really awesome clinical neurologist. So that, kind of shows my love for neurology, since I named my dog after a neurologist. So I guess, shall I go ahead and get started on the lecture? Time to go? Yes. Okay. Awesome. Well, hello everybody. Like they said, I'm going to be starting back at WVU. That's where I did my residency. But I took 2 years to go down to University of Florida for fellowship and just finished up this month. I was studying movement disorders, got a lot more experience in neuromodulation, which I'm going to be talking to all of you about tonight. And there's a little fine print to the title of my talk. Let me see if I can get it to forward. There we go. It's neuromodulation and some things I've learned along the way. So there will be a couple stories I'm sharing as well. So first of all, well, I guess before I even get started, if any of you guys have questions, don't understand what I'm talking about, need some clarification, please feel free to, I guess, do the virtual chat thing or raise your hand or something. I'm happy to clarify. And yeah, so we'll get started. Neuromodulation, what is it? Why do we care? So I'm a clinical neurologist, so I'm going to be talking mainly from a clinical neurology perspective. The really cool part of neuromodulation is there are a lot of different fields involved. So there's a lot of basic science, animal models, physiology, as well as engineering for the hardware and software components of neuromodulation. So it's a really collaborative team approach when we're looking to improve quality of life for patients. Now, interesting with clinical neurology is there's all these different things, and this is just kind of large categories. There's a lot more than what's on this slide here, but things like stroke, which is kind of like a heart attack of the brain. So there's a blockage in a vessel in the brain, and then the blood can't get to that part of the brain. That brain tissue dies and leads to an injury. Or something like multiple sclerosis, which is an autoimmune disorder where basically your own cells are being attacked by yourself. And you can think of neurons, the cells in the brain, as kind of electrical wiring, and they are all housed in like insulation. So just like an electrical wire is wrapped in insulation, your neurons are wrapped in something called myelin, and multiple sclerosis attacks that myelin, destroys it, and then the electrical signal can't get through, and you have some neurologic injuries. So similar things with all of these. What happens is for many of these, there are no cure. And so it can be very frustrating to work in a field where there is no cure for a lot of the things you see. And so neuromodulation becomes this really amazing tool that we can use while it isn't a life as normal as expect. We're going to get into the theory of neurogenesis. Let's see here. So this guy down here, his name, Santiago Ramon y Cajal, very, very famous neuroscientist. What is he famous for? Well, he did all of these stains of neurons, which are really beautiful and artistic in some ways. But why do I bring him up? Well, he studied all of these neurons, and he said in the adult centers, the nerve paths are something fixed, ended, and immutable. Everything may die, nothing may be regenerated. Well, this is a really bleak outlook on neurologic disease, right? Because if you have that stroke and the brain tissue dies, or you have an autoimmune disease and the myelin is attacked, then he's basically saying, well, too bad, that's it, nothing more we can do to help you out. And for a long time, that was the prevailing theory, that once there is an injury to the brain, the spinal cord, the peripheral nerves, that's it. There's nothing more we can do to help you. And you see kind of in history, patients who had strokes or other devastating neurologic illnesses, oftentimes they did not go through rehabilitation. Treatments were not tried for them because it was assumed, well, once there's an injury, there's nothing more we can do. But turns out that's not the case. And so here's a little story for you guys. So I want to highlight these two researchers, Pasco Rahik and Elizabeth Gold. So there's a really interesting kind of competition between these two about trying to figure out if neurogenesis, the development of new neurons after an injury, if that's real or not. So this guy back in the 1960s, he did this really cool experiment. He put an isotope marker into rhesus monkeys, looked at slices of their brain at different stages, and concluded that no, neurogenesis is not possible. He supported what Santiago Ramon y Cajal previously stated. And that was the prevailing thought for a long time. But then some other researchers throughout the 60s, 70s, and 80s started to see that, maybe that's not the case. Maybe neurogenesis actually does exist. And if that's the case, then maybe we can help patients after they've had a neurologic injury. So it was really the breakthrough research of Elizabeth Gold in 1998. She was able to essentially prove that neurogenesis was occurring in the really primitive structures of the brain, the hippocampus and the olfactory bulbs. And her work was so compelling that this guy back here actually retracted his work and said, yep, you are right, neurogenesis is a thing. So this is Pasco here, and this is Elizabeth. So they were kind of going head to head on this topic. And I think there's a lot of really cool take-home messages from this. One is to be persistent, like Elizabeth Gold did, and to really follow the research and follow the theories. And just because one thing was apparently proven many years ago doesn't mean that the science might evolve and other things may become apparent later. The other thing that I think is important is this guy knew when to admit that he was wrong. So that's also a good take-home message. But after her awesome work with monkeys, other researchers went on to show that neurogenesis also occurred in the human brain. So exciting things. So what does this mean for clinical neurology? Well, neuroplasticity, a quick definition here, is the ability of nerve tissue to form new interneuronal connections, right? If you have a nerve cell and another nerve cell, the way that they communicate is between those two nerve cells. That's called the synapse. So when you have new synapses forming, that's synaptogenesis. And then when new neurons are being formed, that's called neurogenesis. There's also this kind of cool existential concept called metaplasticity or Hebbian plasticity, which basically says that whatever the previous synaptic activity was actually determines what its future potential for neuroplastic changes is. Now, looking here, the mechanism of neuroplasticity is super complex, not fully understood, but we're going to give you a really quick rundown. As you see here, there's two terms that I want you to kind of store in the back of your memory. So long-term potentiation and long-term depression. These terms we're going to come back to throughout the talk to kind of explain how all these really cool neuromodulation devices work. So when we're thinking about long-term potentiation, that's an excitatory response, and it's increasing neurogenesis and neuroplasticity. We have neurotransmitters going from one neuron, this is one neuron here, to another neuron over the synapse. These stimulate a variety of receptors that allow the calcium to influx in. and that stimulates a variety of protein kinases, going back to biology, which triggers this lateral diffusion of more receptors, strengthening those connections along the synapse. The opposite occurs in long-term depression. So if biology is not your thing and you really want to focus on the take-home message from this slide, just focus on these two terms, long-term potentiation and depression, and you'll hear more about those later. Now, before I go on, there's a quick disclaimer. We don't know the precise mechanism of how most neuromodulation devices work. So this can seem a little alarming because a lot of neuromodulation devices involve surgery, where we're actually doing brain surgery or surgery to the spinal cord. And we don't know how they work. We have some ideas, we have some theories, we're working on it, but we just know that they do work, and there's really profound clinical changes once these patients have these devices implanted. So without further ado, let's start the neuromodulation part of the talk. I'm going to start with something called non-invasive brain stimulation, or NIBS. And this is kind of my main research area. What I like about non-invasive brain stimulation is it does not require surgery, but you can still have really significant clinical impacts. So the first device I'm going to talk to you about is called transcranial magnetic stimulation. What it uses is a magnetic field. So there's this coil right here that sits above the patient's brain over their skull. And basically the magnetic field induces an electric current in the brain. How we do the mapping, I'm going to show a video that kind of demonstrates it, because I think that's a little bit easier to understand. So I'll show the video first and then kind of explain what's going on.
You ready?
Yeah, I'm ready. Right, I think the answer is quite difficult, isn't it? Bring on the next test.
So if you want to go ahead and start doing some finger touches
touch
again.
my nose. I can keep this up for hours.
Excellent.
Yeah, so just keep going.
It's pretty dramatic.
That's quite dramatic. I was in some stage of poking my eye at that moment. I thought.
So could everyone hear the sound okay? I'm not sure if that was coming through or not. Well, hopefully yes. So in terms of what's going on in this video, they're using the coil to stimulate the brain. How do we know it's working? Well, it's actually being held directly over the motor cortex. Now, the right side of the brain controls the left side of the body and vice versa. So in this video, they're stimulating over the left side of the brain, the motor cortex, and he's moving his right arm around. Every time there's a little bit of stimulation, you hear that tapping sound, and it interrupts his motor coordination. And so there's a tap from the TMS machine, and then all of a sudden his arm is not functioning properly. Now, that is not the point of the TMS device. The point of doing the motor cortex mapping is to figure out the appropriate threshold for which to set the TMS coil. Then we move the coil to a different treatment location to give the patient their treatment. So I think this diagram kind of helps explain mechanistically what's going on, or at least what we think is going on. So there's low frequency and high frequency stimulation. So low frequency just means that the pulses, that clicking sound that you heard in the video, are delivered slowly, and high frequency is going to be more than 10 hertz, so more quickly. So whenever you have low frequency, that's inhibitory stimulation. And what we think is happening is the electrical stimulation that's coming from the coil, or induced from the coil, is essentially inducing long-term depression, back to that neuroplasticity slide. Whereas high frequency stimulation induces long-term potentiation. It's excitatory. So you might want to use a low frequency protocol if you have something that is a hyperkinetic state. So if you have a tremor, that's kind of a hyperkinetic disorder. And we do a long-term depression or inhibitory stimulation, and it can calm the tremor down. High frequency stimulation, if you have a disorder that is kind of, you can think of hypokinetic or slow, something like depression, you can excite the brain in a particular anatomical point and potentially improve major depressive disorder. There's other types of TMS. It's not just high and low frequency. There's also something called theta burst stimulation. So each of these thick marks right here is really a triplet burst in a theta rhythm. And all that means is that you're getting a triplet burst. Let's go every time you see one of these markings. If you deliver that continuously, it's the same thing as high frequency stimulation. It's excitatory, or I'm sorry, it's the same thing as low frequency, it's inhibitory. And if you do intermittent, meaning you do a bunch of pulses, do a pause for a couple of seconds, and then a bunch more pulses, that is similar to high frequency stimulation, which is excitatory. So what is TMS used for? I mean, this is cool. It's a cool device. It's cool to see how it kind of interrupts your motor movements when you're doing the mapping sessions, but what is it actually used for? Well, it's only FDA approved for major depressive disorder, anxiety, and OCD. And anxiety and OCD were just really recently approved. A different version of TMS called single pulse TMS is also approved for migraine, and we'll go over all of that. So although there are only a couple things TMS is FDA approved for, it's being looked at as a research tool in a variety of disorders. And this is from a particular manuscript that covers all of the uses up through 2020 that TMS has been used for. So you can see there's a lot of potential here. It's really exciting because if we can use this device to help patients with all these different types of disorders, that could be really, really exciting. So another quick story for you guys. How did I get into TMS? Because it seems a little kind of random, right? So my story is that when I was in residency during my second year of residency, I was thinking, I'm interested in post-stroke depression. That seems really interesting to me. Why was it interesting? Well, what I was finding in patients who had strokes, it didn't seem to matter if they had the really large strokes where their whole arm and leg were affected, or if they had really small strokes and had hardly any deficits at all. There seemed to be a large percentage of patients who were depressed. And so it got me thinking, I wonder if there's something we can do to help this patient population, because the focus is so much on just saving people who have strokes. And then we kind of forget about how to rehabilitate them and treat their post-stroke complications. So I'm just, I'm a second year resident, just kind of chilling at a headache conference in California, because that was a fun place to go. And at this headache conference, they talked about TMS for headaches, for migraine. And they mentioned that it could be used for depression. And I got really excited because I had been thinking, I'd like to do something. So these are all of my original notes from that conference many, many years ago. And I went up to one of my faculty mentors and I said, hey, I'd really love to do a research project on post-stroke depression. And she said, yep, we'll do something really easy, not a big deal, really easy, straightforward, sure, fine, whatever, send me your research protocol. So I said, yes, definitely, straightforward, easy, yep. I'd like to do TMS for post-stroke depression. It's a 4-day protocol. We're gonna have to convince the FDA to let us do that. We're gonna have to convince the IRB that it's safe and find the funding to do it. So not really actually that easy, but I sent her my research protocol and I didn't hear back from her for many, many days. And I figured she probably thinks I'm crazy and is avoiding, talking to me. But after 4 days, she sent me back an email that just said, this is awesome, let's go for it. And we did. These are two of my mentors and one of my co-residents, and we are learning how to do TMS.
It's a very small movement that I'm doing.
So the reason I bring this up, we were eventually able to present it at a conference and publish it as a manuscript, and helped a lot of patients out, which was really, really exciting. But the reason I bring it up is, as exciting as this was for me, a lot of people as a second-year resident would be told, no, you're just a second-year resident, just wait, wait to do it when you have more time, or wait to do it when later when you're at a higher level of training. The reason I was able to do it is because I had a really awesome mentor. This is Dr. Adcock, who sent me back the email saying, awesome, let's do it. And so I encourage all of you guys to, if there's something you're passionate about, go for it. You don't have to wait, and seek out people like her that are going to support you in your endeavors. Right now, I have expanded my TMS to look at TMS for patients with Tourette disorder, helping with their tics, as well as cervical dystonia. So just as a quick background of what is Tourette syndrome, I think it's become a little bit more well-known on TikTok actually these days, but Tourette syndrome is a neurologic disorder that causes motor tics and vocal tics. These are the most common ones are things like eye blinking, clearing the throat, but you can have some really devastating tics that lead to hospitalizations, neck stabbing tics that can actually lead to neck fractures and spinal cord injuries. So some of these people are really, really devastated by the Tourette syndrome that they have, and TMS can actually be used to reduce those tics. Cervical dystonia, the other project that I'm currently working on, is dystonia is, I think the best example is a writer's cramp. So if you've heard you're writing and all of a sudden your muscles cramp up and you can't write anymore, that is a specific type of dystonia called focal task specific dystonia. But you can have generalized dystonia where it affects your whole body. You can also have cervical dystonia where you have a tilt in your neck, or your neck is kind of fixed, and sometimes you have a tremor too, like that. And TMS can potentially help with that as well. So all of the cool stuff that I'm doing right now is really thanks to my mentor, who I met back in residency, which I think is pretty cool. So the other TMS application that I want to talk to you about is migraine. Usually I would ask, how many of you have migraines, but I can't really see a lot of you. So, but I assume that many people have migraines. It's very common to have migraines. And one of the interesting things is a lot of people have auras before their migraines. I don't know if anyone has an aura before their migraine. What is an aura? It can be many things. The most common is a visual disturbance, which can look like these colors or these kind of weird patterns in your field of vision. Now, the cool thing about an aura is we can actually map it. If you stick someone into an fMRI machine and look at their brain lighting up when they have an aura, you can actually see it in the occipital lobe. And then something called cortical spreading depression happens, where it spreads to the rest of the brain and eventually causes the pain part of the migraine. So the idea behind TMS for migraine is, this is the portable device. You use it at home. When you have the aura, which is kind of your warning sign for a migraine, you hold it up to the occipital lobe, where we know that that aura is forming in the visual part of the brain. You put that back there and you deliver a pulse of TMS to your own brain. And the idea is it just disrupts that cortical spreading depression and stops the pain aspect of the migraine. So it's pretty cool. And this is actually an FDA approved indication. The other type of non-invasive stimulation that I want to talk to you guys about is transcranial direct current stimulation. So just as a refresher, TMS is this coil that is inducing an electric current in the brain. Direct current stimulation, as the name implies, is there are direct electrodes connected to the scalp. What is different? Well, if you have anodal direct current stimulation, that is similar to high frequency or intermittent theta burst stimulation, which is excitatory And remember that mimics long-term potentiation in the neuroplastic realm. Whereas if you use cathodal direct current stimulation, that mimics the TMS aspects that also induce what we think induces long-term depression. So it's inhibitory stimulation. So very similar concept, just a different way of delivering the stimulation. And this is just a picture to kind of demonstrate how that would look in a setup and kind of the brain readings that we're looking at back there. So you're probably sitting there wondering like, well, this sounds awesome, but like, why is it only FDA approved for a couple of things? Like, shouldn't we be using it for Tourette's and cervical dystonia and Parkinson's and gait freezing and post-stroke things? Yeah, I think we should. But there's a reason why it's not. So there have been a lot of cool studies. I'm just going to highlight a couple of things that have had positive results. Upper arm paralysis following a stroke, or post-stroke hemiparesis, which is when the whole entire body has been affected by a stroke. That it has shown that different types of non-invasive brain stimulation has given back function to these patients, especially when it's combined with really aggressive rehabilitation like physical therapy. Aphasia, what is aphasia? That is dysfunction in your speech. Typically, it's following stroke. So if you have a stroke to the language center of your brain, you're not able to produce speech anymore, or sometimes you cannot understand speech, or sometimes both. And you could imagine that would be very difficult to rehabilitate if you can't understand or communicate with someone else. You can also have aphasia as a primary progressive form, which is basically a type of Alzheimer's that just affects your speech. And non-invasive brain stimulation has shown improvements in speech. Parkinson's is a neurodegenerative movement disorder. If you've seen someone who's kind of flexed over, walks slow, the hallmark features of Parkinson's are slow walking, the pill rolling tremor at rest. At rest is important. These patients, they also have something called freezing gait, which is really, really difficult to treat. You can think of freezing gait as, if you imagine an Energizer bunny, you wind it up, it starts going forward, and all of a sudden it kind of glitches and it doesn't go forward anymore. That's freezing of gait, and it can be really challenging for patients with Parkinson's. You can imagine they have falls, they can have really significant injuries, and they can't be independent because of it. So non-invasive brain stimulation has helped with that. There's also this really kind of crazy concept known as neglect, most commonly happens after stroke. So if you have a stroke to the right side of the brain, in the parietal lobe, that is telling you how you see things in space. And so for some patients following a stroke, they just forget that the left side of the world exists. They just don't realize that it exists anymore. So if you ask them to draw a clock, they'll draw the circle and they'll put numbers 1 through 12 all on the right side of the clock. It's as if they don't see that the left side exists at all. So we're going to move here to our next part of the talk, is invasive neuromodulation. This is the most exciting of the modulatory field. And so we're going to start with something called spinal cord stimulation. Spinal cord stimulation was traditionally used for patients who pain, intractable back pain. And they put a spinal cord stimulator in to help them. In terms of how we can use it for neurologic illnesses, the thought was, well, if patients have a spinal cord injury, meaning that below the level of the spinal cord, they can't move and they can't feel anymore, and this is the grading scale, what if we stuck a spinal cord stimulator in and tried to make them walk again? And they found that they actually could, which is really impressive. So just so for your own reference, if you do not have a spinal cord injury, that means you're normal. If you have a spinal cord injury that affects motor and sensory, you're A or B. If it doesn't affect motor, just affects sensory, that's C or D. And you might think, well, if it's just sensory, that's not such a big deal, but that's still a big deal. So if you imagine when you're walking, your brain needs to know where your foot is in relation to the ground, something called proprioception, where it can tell where it is in space. And if you don't have that, you might look very clumsy, and you might not be able to walk at all because your sensation is not intact. So it's still a very devastating consequence of spinal cord injury. So this is the spinal cord stimulator. These are the electrodes. These wires come through here and connect to the battery, which delivers the stimulation. So I'm going to show a short video of one of the first patients to have this done.
It's been 5 years since a snowmobiling accident left Jared Chinook paralyzed from the chest down. And it's been 2 years since a multidisciplinary team of researchers implanted an epidural electrical stimulation device in his spine, along with intense physical therapy, to help Jared use his legs again. Since then, this avid bow hunter has reached and exceeded several milestones in a clinical research collaboration between Mayo Clinic and UCLA.
Like my sitting balance and stuff has gotten a lot better. I can shoot my bow a lot better because I'm able to have more trunk support. My standing's gotten a lot better. I can stand unassisted, I would say, with the walker, but still have my hands on, but lightly. Walking on the treadmill has gotten a lot better and easier. All depends on how much my arms can take too. And then average bout, I mean, I can walk, what, 30 feet or so, or more probably. Definitely some distance to it.
On his best day, he was able to take steps, with some help, more than the length of a football
So
field.
this is very exciting, right? This is a patient who was unable to walk, and with the help of really, really aggressive physical therapy and a spinal cord stimulator, he does have some control over his walking capability. So that's huge, right? A similar study at the University of Kentucky looked at 4 patients who also had complete spinal cord injuries, and just some things I want to point out about the study is they did 278 locomotor training sessions over 85 weeks with stimulation. So this is not just like a magical, we're going to put the spinal cord stimulator in and they can walk again. It still takes a lot of physical therapy and retraining those neurons to kind of develop those neuroplastic changes that we talked about. The other interesting thing about this study was that the patients couldn't perform these actions, like the walking, when the stimulator was off. So that's a little unfortunate because you want them to still be able to function whether or not the stimulation is working perfectly. But I'm just going to show you a couple videos of the patients that were in the Kentucky study.
Participant 3 is a man with a motor complete, sensory incomplete traumatic spinal cord injury at the 5th cervical level, walking over ground using epidural stimulation while holding moving parallel poles for balance, but without manual assistance after locomotor training. Walking occurs only while epidural stimulation is turned on and while the participant consciously intends to walk. Participant 4 is a woman with a motor complete, sensory incomplete traumatic spinal cord injury at the 1st thoracic level, more than 3 years post-injury, walking over ground with epidural stimulation without assistance using a roller walker. Has undergone locomotor training with epidural stimulation.
So the last video I'll point out, the reason they're showing this is he has good truncal control. So imagine if you have a high enough spinal cord injury, you can't even sit by yourself. So imagine going from not being able to sit down by yourself and stay upright to being able to sit and use your arms to function. That can make a world of difference, on top of being able to give patients the ability to walk again. So a team of researchers said, well, that's great and all, but how about we make it even better? You know, like it's a little frustrating, right? If we put the spinal cord stimulator in, but we don't really know how it's working or which muscles would really be the best for that particular patient. And so they tried to do this thing called biomimetic stimulation. I'm going to show a couple of videos. This is still an active area of research, but this is some of their preliminary subjects that were in the study. Oops, clicked off of that one.
Recording kinematics and muscle activity.
So let
Aimed at
me.
delivering electrical stimulation.
So this is a healthy individual, and they use an EMG, which are stickers on the muscles, to record the muscle activity. Then what they do is they map that muscle activity to the different neuron roots, or what they call motor neuron pools in the actual spinal cord. So they know which areas are lighting up in a healthy individual based on that muscle activity. Then what they do is for the surgical procedure, this is how they are kind of opening the back up. They insert the spinal cord lead, the electrode lead goes into the spinal cord. And then what they do during the surgery, which is pretty cool, they deliver stimulation to the area where the electrode is. And they say, oh, there was no muscle movement there. So we're going to adjust the electrode to a different level of your motor neuron pool. We're going to stimulate that. And oh, look, after the stimulation, your leg moved. So now we know we're in the right spot. So once they get that information, they put the spinal cord stimulator into the patient. And you can see when there's no stimulation, he has a gravity assistive device because he can't bear his own weight, but he can't walk even with gravity assistance. Then when they turn the stimulation on, you can actually map the motor neuron pools in the spinal cord and what is being stimulated to the muscle activity in his legs as he's walking. So really, really fascinating stuff. The other cool part about this study is that even after the spinal cord stimulator was turned off, after months and months and months of using it, we see that there is some retained benefit even without stimulation. And so these are a couple more videos showing the patient walking across ground with a gravity assistive device again. You can see a lot of dragging in that left foot. This is before he had surgery, so he's partially complete spinal cord injury. Now, a couple of important differences after he has rehabilitation and the stimulation has already occurred, but they have turned the stimulation off. So he currently has no stimulation turned on. Let me go back to that because I think this is really powerful. He no longer has a gravity assist device, right? He's bearing his own weight and he's just using a couple of canes to walk across the ground. And this is all because of months and months and months of rehabilitation with spinal cord stimulation. But now he's able to function without stimulation. He can do even better with stimulation. So this company partnered with some researchers and said, you know, that's great and all that these patients can walk again, but, you know, it's kind of frustrating, right? If you want to go ride a bike or you just want to stand somewhere, you know, it would be nice if you could like change the stimulation settings on kind of a, you know, a snap your finger kind of basis. And that way you can have your program that controls bike riding and your program that controls walking and all these different programs, right? Oops, let me go. So what they did is they took kind of like an Apple Watch type of device and they said, okay, you're going to have a wearable programming device and you can decide to start the stimulation yourself. You can switch to the cycling program or the standing program or the sitting in a chair program, and the stimulation will change depending on what activity you want to do out in the real world. So this is taking something from all the way from the beginning to using it in really practical, exciting applications. What is really exciting to me and was just presented at a research conference is they want to take biomimetic stimulation to the next level. If that Apple Watch device wasn't enough for you, those researchers said, no, we're going to do even better than that. we're going to stick cortical electrodes, so sticking electrodes in the brain to monitor what your volitional movements will be. And so these cortical electrodes that are sitting in the brain will monitor what your intended movements are. And if you intend to go cycling, or you intend to stand, or you intend to walk, those cortical electrodes will relay a message to the spinal cord stimulator, and it will automatically change the program for you. So it's almost as if you have regained normal function. You don't have to go through a button anymore. If your brain is thinking that, the program changes for you. Still in the early stages, but really, really exciting, right? These are patients who before their options were extremely limited, and there's some really exciting things on the horizon. Another brief story for you guys. Does anyone know who this guy is, or who said this? If you do not expect the unexpected, you will not find it, for it is not to be reached by search or trail. And I'm sorry, I can't actually see the chat screen. So some of you may have gotten it, but we're going to go on and say it's Heraclitus, so an ancient Greek philosopher who was famous for many one-liners, including expect the unexpected. And the reason I bring this up is when they implanted a Parkinson's patient with a spinal cord stimulator for pain purposes, they said, oh, hey, look, his walking is a lot better. So kind of a serendipitous finding that led to a bunch of clinical trials in which they looked at spinal cord stimulation for that freezing of gait, so making that Energizer bunny walk the way it's supposed to walk. So just to compare, this is a Parkinson's patient without spinal cord stimulation, and this is the best that they can do on medication. And you can see these are their footprints as they're walking across the room, and they keep freezing, they keep pausing. Whereas at their best setting on a spinal cord stimulator device, they can walk essentially normal. Even on the worst setting for spinal cord stimulation, they have a little bit of freezing at the beginning, but once they get going, it's essentially a normal gait. So that's with the absolute worst setting on spinal cord stimulation. Still much, much better than on medication and no spinal cord stimulation at all. So just like kind of the serendipitous discovery of penicillin, it's important to kind of keep your eyes open to kind of these other little nuances when you're treating patients or looking into different things. So the next type of invasive stimulation I'm going to talk about is responsive neurostimulation, or RNS. This is a really—
Jessica?
Yes.
We have like some question on the chat, like before you go into the next topic.
Sure.
Yeah, it was from Timothy. Yeah, I don't know if you want to do a question.
Yeah, I'm happy to.
Oh, I just read.
What was that?
The question was, so there are like prosthetic nerves. It was just on the last topic.
What is, oh, prosthetic. So they're like prosthetic nerves. Oh, yes. So that's an interesting analogy. It's not quite a prosthetic nerve, but it's retraining the injured nerves. It's all coming from this neuroplastic concept, which is that the brain, the spinal cord, the peripheral nerves can relearn how to do things. So it's not necessarily that you are like putting a prosthetic limb on as kind of a new nerve, but you're retraining existing nerves using the neuromodulatory devices to be able to function more normally again. Does that answer your question? If not, I am happy to kind of clarify further. But
we can jump into the Q&A session after the lecture and
Sure.
clarify further if necessary.
Awesome,
Yeah.
awesome.
Like Timothy say yes. So.
Yes. Yeah, if you have questions, happy to answer them, but I will kind of get through a couple more of these slides. I know we're running a little short on time, so I'm gonna try to do my best to get through all of them. We'll see how that goes. So RNS, really cool device that is used for epilepsy. What is epilepsy? It's a predisposition to having seizures more than the regular population. So any one of us could have a seizure if we put enough stress on the body, or on the brain, I should say. But people with epilepsy have a tendency, even without extra stresses on the brain, to have seizures. What is a seizure? It's basically an irregular firing in the brain. It oftentimes will start focally and then generalize. When it generalizes, it often turns into something called a tonic-clonic seizure, which is probably the one that most people are familiar with. That's where you kind of stiffen up and then you start to shake all over and lose consciousness. So with responsive neurostimulation, that's approved for focal epilepsy. It's basically a brain pacemaker. And you can see that patients who have frequent seizures have really remarkable reductions in their seizures once this device is in place. So this video does a good job demonstrating how it works. It's really, really cleverly designed. So this is the IPG, which delivers the stimulation. It's basically the battery goes drilled into the skull. And then these electrodes are placed. We previously, during a previous surgery, map where the seizures are coming from. So the electrodes are placed specifically where the seizures are coming from. And so what you see in this strip here is it's detecting abnormal electrical activity in the brain. And so it delivers some stimulation and it causes that seizure to end before it begins. Another abnormal rhythmic stimulation here. We better deliver some stimulation and get the seizure to go away. So it acts as a closed loop system, meaning that we don't have to do any specific programming. The RNS system acknowledges the seizure, takes care of them, and stops the seizures from happening before they generalize into those tonic-clonic seizures. So really, really cool. The next invasive type of stimulation that I'm going to talk about is deep brain stimulation. And this is what I have the most experience with clinically. So it's a really cool device. What is it? Deep brain stimulation goes into the deep brain structures, as the name suggests. It goes into very specific targets. There are specific nuclei in the brain that we know are kind of responsible for causing diseases like essential tremor and Parkinson's disease. And so the idea is, here's the electrode in the brain. A wire runs down to the pulse generator, which is basically the battery. The way that surgeries are done for DBS are really fascinating. So we look for the structure on some sort of imaging, like a CT or an MRI, and then we determine the coordinates of where we want to put the lead based on that image. There's a frame that goes around the patient's head, and those coordinates correlate with this frame that the neurosurgeon uses so that they can get the exact targeted place. However, sometimes we don't get exactly right. Sometimes we're a couple millimeters off. So how do we figure out or fine tune and make sure it's in the best place possible to get the best clinical outcome? Well, we actually let the patient be awake for the surgery. That sounds really scary, right? This is brain surgery and the patient's awake. Why is that? And the reason is twofold. One, we do something called microelectrode recording, also known as MER. And as the electrode is being pushed through the patient's brain to its intended target, we can actually hear the different structures of the brain that we're passing through. So imagine this, you're in the operating room, you're on the table, you're listening to your own brain. Pretty cool. So we can tell which nuclei we're in based on the sound that we're hearing. So we collect a whole bunch of data that way, and then we also look for a clinical response. So this is usually kind of my role as the neurologist. I'm testing their clinical response and seeing if there's benefit or if we need to move the lead in any kind of other position. So DBS is approved for essential tremor, Parkinson's disease, dystonia, and epilepsy, a different kind of epilepsy than the RNS was approved for. I want to show you a couple examples of the surgery. So this is a patient who's obviously awake and playing guitar. Why is he playing guitar during the surgery? That seems a little like counterintuitive. The reason is this patient has something called musician's dystonia. So if you are a guitar player and you make your livelihood based on playing guitar, but you get kind of that writer's cramp sensation in the hand that is playing the notes, then that could be really devastating. And so the DBS is approved for dystonia, including musician's dystonia. So what they're trying to do is look for inducing the dystonia, inducing the musician's dystonia while he's playing guitar, so that they can figure out if they're in a good target spot in the brain. This is a really fascinating before and after video. Okay, so this patient has really, really severe tremor predominant Parkinson's. You can see that tremor is really, really severe, and his DBS is currently turned off. So this is before stimulation. Now the programmer, fast forward here a little bit. Oops, fast forwarded too much. So he turns it on, and just like that, the tremor goes away because of the deep brain stimulation. And you can imagine going from this to this is got to be life-changing. So this is another example of Parkinson's disease before and after stimulation. You can see the motor control that you get after deep brain stimulation is really fascinating. I'm going to skip this video for time. So how do we determine, how does DBS work? How do we help these patients? These are a variety of leads that we stick into the brain. This is the device that actually gets placed in the brain. Depending on the company, you have a bunch of different options. Each of these black areas is the contact that actually delivers the stimulation. There's a couple of different things that we can control for. So we can, I'm going to use a little example of the lighthouse because I think this analogy is kind of the best way to explain it. So if you imagine a lighthouse, like on the coast somewhere, you imagine this lighthouse is emitting a light, and the frequency, it's the amount of times that you see the light go around. The pulse width is the amount of time that when you're looking at it, you see the light sweep around. And the amplitude is the amount of light, the strength or intensity of the light. So these are the three things that we have to control on our little programmers. And depending on how we change them, that also depends on the area of the brain that is being stimulated. So there's a lot of really awesome research in the works for DBS. I don't have time to go over all of it, but I'm going to highlight a couple of things. So DBS in Alzheimer's disease, obviously an incurable neurodegenerative disease that affects many, many people in the world. The first case of DBS for Alzheimer's was actually in 1985. But there have been much more done recently. This is just showing that we think DBS is increasing acetylcholine release, which is a neurotransmitter thought to be low or deficient in Alzheimer's. But the mechanism is much more complicated than that, and we'll kind of skip over that for now. One of the really fascinating articles, I think this was a New England Journal of Medicine, was that based on the increase in the voltage, so going from 7 volts, 8 volts, 10 volts, the memory became much more clear in the patient's mind. And they had a whole case series where this was the case for all of those patients. So really fascinating. More work needs to be done in this to kind of determine how this is working. There's also this really awesome concept of closed loop DBS. You can liken closed loop DBS to automatic versus manual driving. So in manual driving, we are the programmers, we are determining the stimulation parameters. kind of like stick shift, whereas automatic driving, it determines which gear it needs to shift into and when. This gets into what we are kind of looking at mechanistically. So there are different frequencies that neurons oscillate at. And for some reason, the beta oscillation, this frequency, beta frequency, seems to be associated with the types of symptoms we see in Parkinson's disease. So the slowness, the stiffness, the tremor. And so the idea is if the DBS lead can kind of look around for that beta oscillation, if it can sense that beta oscillation, then it can turn on and stimulate when you're having symptoms, and it can turn off when you no longer are having those beta oscillations. This is an example that we just recently did at University of Florida. This is actually a patient with essential tremor, not quite using the technology to look at oscillations yet, but what it's doing is it's looking at the EMG, which are these electrodes applied to the muscle. And so here's the EMG activity. And so when there is no EMG activity, the stimulation automatically turns off. But when he goes to move his hand, the stimulation automatically turns on. And researchers are now looking at ways to take this from an EMG signal where you're using a wearable device to directly looking at brain signals to help do that. So we're going to pause that there. I know I'm running a little over. This is the last portion of the talk, so I'm going to kind of just quickly go over brain-computer interfaces because I think they're really, really cool, and then we'll get to the question and answer. So brain-computer interfaces. Going back to the prosthetic question,
So this was
there is the ability to take these really kind of wild prosthetic devices that have sensory capabilities. And in this patient, she lost her arm in kind of this really random accident where a raccoon bit her and it got infected and they had to amputate her arm. So what they've done is they've taken the nerves and they've mapped them to different parts of the prosthetic arm. And then over a series of training sessions, they train which parts of the arm here kind of map to the fingers on the prosthetic, so that when they touch different areas of the prosthesis, the patient can actually sense that touch. And in her brain, it is interpreted as touch in her own hand, even though she no longer has a hand. So this is really revolutionizing the way we think about neuroprostheses, because like I said before, if you don't have sensory capability, which previously prosthetic limbs did not have sensory capability that could go directly to the brain, then you are unable to function normally. I'm also going to briefly mention NEURALINK, which is kind of this really wild concept of using, let's see where we are.
Who had a Neuralink placed in each.
These brain devices to control things. So this is a monkey that he's getting a treat, a banana smoothie through this straw here, and he's using the joystick to control this game, and they're training him to use his mind to control the game. So if we go down a little further, you see he's still playing this game, right? And he's still moving the joystick, but the joystick is not connected anymore, meaning he's just moving this joystick out of habit, but it's his brain that is actually controlling the game. How does this work? Because that seems really wild, right? So there's a lot of neurons that are being processed, which you can see in blue and red here, blue for up, red for down. So the neurons that are being processed when blue is more, it denotes an upward signal, which is, you can see here is upward, and the upward and downward deflections translate to how he's able to play this Pong game. So really, really fascinating. But I want to do one last interlude. Does anyone know who this is?
Jeff Goldblum.
Yes. You know what character he played?
Some guy
from
from
the name, but it's
Park.
Jurassic Park.
Yes, yes. So this
Edition.
is Ian Malcolm. And his famous line from the original movie or book is, "You were so preoccupied with whether or not you could, you didn't stop to think if you should." And this is what he was talking about. So my kind of word of caution is there are a lot of ethical questions for a lot of these devices, not just Neuralink, but I think that has some of the most ethical questions since it's the newest technology. But for any of these devices, what is normal, right? We are constantly talking about trying to make patients normal again, but what is normal? And where's the line between restoring personhood and augmenting human nature? And does hardware and technology, does that make you more human, or does that actually strip you of your humanity? And how do we decide who benefits from this technology and who is harmed by it? Now, that is another topic for another day. You could do a whole series of lectures on just the ethical questions of neuromodulation, but I want to throw it out there because I think it's important to always step back and think about, hey, this is really cool and this can help a lot of people, but let's think about kind of what we're doing here from the larger picture about things. So in conclusion, be persistent and don't be afraid to admit when you're wrong. Surround yourself with people who will support you, and don't be afraid to go after your dreams, even if you're not at the quote right level of training. That's always the right time to go after your dream. Expect the unexpected, in the famous words of Heraclitus. Don't make dinosaurs. Good life advice for anyone. And do what you love, because if you do what you love every day, then it won't feel like work. It will feel like something that you were born to do.